So very closely related to the hypersensitivity reactions are the autoimmune diseases. In fact, the type 2, type 3, and type 4 hypersensitivities are very much intertwined with a lot of autoimmune diseases. So if you go back and look at a lot of those tables, the diseases associated with those type of hypersensitivities are very commonly autoimmune diseases. Now, autoimmunity is extremely complicated. There's not one major sort of factor that we can say is the sole factor in causing autoimmune disease, okay? However, the key factor, the one thing we do know that's common among all autoimmune diseases is that there's an issue with the immune system not being able to differentiate self from non-self, right? So the immune system is now recognizing self-antigens as foreign antigens, becoming activated, and really launching an all-out immune response, but against self-tissue. So this autoimmunity initially results from some sort of a failure of central tolerance. As remember, our T cells should be educated in the thymus, and B cells are selected and or educated in the bone marrow. And we're going to talk about some of those proposed mechanisms, but again, have to keep in mind that autoimmune diseases are really complex and it's never just linked to one specific factor. Self-reactive lymphocytes that somehow escaped selection in the primary lymphoid tissue are usually subjected to different mechanisms of tolerance actually in the periphery, okay? And there are three primary mechanisms that induce peripheral tolerance. One is anergy, and we'll talk about that in just a minute. Another is deletion, which should have happened within the primary lymphoid tissue, like the bone marrow or the thymus. And the other one is suppression. Remember when we were talking about the different T helper subsets, the regulatory T cells. The job of the regulatory T cells is, again, to suppress some of those T cell mediated diseases. And in some people, those regulatory T cells aren't functioning, which allows those diseases to go sort of unregulated and uncontrolled, which can also lead to autoimmune disease. All right, so we're going to take a look at B cells first. We talked a little bit about B cell selection. We know that this happens in the bone marrow during the immature B cell stage. But should cells get released into the periphery that are potentially reactive to self, in the absence of T cell signaling, they can become anergic, which of course means that they're non-functional. And these anergic B cells can express high levels of IgD on their surface, and that is one of the signals that sort of excludes them from the secondary lymphoid tissues. These anergic B cells then cannot receive the signals necessary for survival. Remember, B cells that have not come in contact with the foreign antigen only have a very short lifespan unless they come in contact with the antigen that they're specific for. So these anergic cells would not receive the signal necessary for survival, and they would undergo apoptosis. B cells have an additional mechanism by way of inhibitory receptors that can be engaged or ligated when we bind to self-antigen, and that can help to suppress their activity, okay? T cells, very similar to sort of what we talked about with B cells that recognize self-antigen, again, in the absence of those co-stimulatory signals, can also be turned off or deleted in the periphery. So with T cells, anergy is the result of a breakdown in the T cell receptor signaling itself, again, not receiving perhaps the right co-stimulatory signals, or the binding of inhibitory receptors. Now, these inhibitory receptors like CTLA4 and PD-1 are really big right now in cancer immunotherapy. Because if we inhibit the inhibitors, we can keep the immune system sort of working, and that has done really sort of some wonders in treatments of different sorts of cancers. So, deletion of these self-reactive T cells is due to apoptosis, usually by activation, and you guys know this already, of the caspase signaling pathway or Fas signaling pathway. And as we mentioned earlier, we don't want to forget about the regulatory T cells. So T cells can also be suppressed by these regulatory T cells, okay? A lot of these are generated during selection. Remember, as sort of a last resort, if a T cell doesn't pass selection, it has one last chance to survive, and that's to become a regulatory T cell. But some can also arise in the periphery as well. And these are going to be really important in helping to maintain proper homeostasis and prevent autoimmune disease. If you remember back to when we were talking about the regulatory T cells, we know that Tregs secrete lots of IL-10 and also TGF-beta. And this does a really nice job of really suppressing, especially that Th1 response that is going to give us a nice vigorous macrophage activation, etc. So that is going to be really important in helping prevent certain autoimmune diseases. CTLA4, which again is the inhibitor, it binds to B7. It looks a lot like CD28, but CD28 is the activation marker. CTLA4 is the inhibitory receptor. And this inhibitory receptor is expressed at really high levels on regulatory T cells and can sequester that co-stimulatory B7, which again could be used to activate T cells. So that CTLA4 is an important inhibitory molecule on T cells, in particular regulatory T cells. As we mentioned, autoimmunity is pretty complicated. So the development of autoimmune diseases is not generally linked to one particular factor, but usually a combination of both genetics, as we're going to talk about with HLA here in just a minute, environmental factors, so exposure to different things, hormones can also be a factor. So again, not just typically one thing that triggers an autoimmune disease. Genetically, though, we know that the strongest associations are with the HLA genes, and these HLA genes, of course, are your MHC molecules. We also see certain contributions to autoimmunity in non-HLA genes as well, but again, the strongest association is going to be within typically the class 2 HLA genes. So this table gives you a very short list of some of the HLA markers or alleles that are linked to autoimmune disease. Again, this is not an exhaustive list, but this is sort of the most commons. One of the things I just want you to notice as you're looking at the different HLA types is that most of them are class 2. So there are way more class 2 associated HLA diseases than there are class 1. HLA-B27, though, is sort of a big one as far as class 1 associated diseases, okay? I don't necessarily really want you to memorize this table, but know, of course, HLA-B27, and you definitely want to know that that's class 1. And the diseases here, especially ankylosing spondylitis and reactive arthritis, those are going to be pretty high yield. But in general, the DR2, 3, and 4, not probably an allele that you would want to get sort of in a lottery of HLA alleles, because a lot of those have HLA autoimmune disease associations. Okay, so again, don't memorize, just sort of be familiar with. But again, that B27 is one that if I was going to memorize that one, I probably would pick the B27. As we mentioned already, autoimmune diseases are fairly complicated, so we know that it could be a combination, and is often a combination, of genetic factors and environmental factors. Infections are part of those environmental factors. Tissue injury, exposing antigens that were usually considered as sort of sequestered antigens that the immune system wouldn't typically see, that now there was some sort of trauma or inflammation and those got released, could also be an inciting factor in the development of an autoimmune disease. Okay, But again, what we see in these diseases that are really very complicated is that a lot of the time, if we have an autoimmune reaction to one tissue, we can actually see other tissues involved as well. And these diseases tend to be very chronic and tend to be progressive. You will also probably have lots of questions as you're going through this, like, well, I don't understand how this happens. I don't understand, why, we're starting with a type 2 hypersensitivity and then it goes to a type 4. There are a lot of autoimmune diseases that are mixed hypersensitivities, right? So they can be a 2, 3, and a 4, or a 2 and a 4. And we want to leave that complicated stuff to the rheumatologists, okay? For step 1, you need to focus on how the disease starts, okay? Is it by antibodies binding directly to a cellular tissue? If it's yes, ask yourself, is it damaging the tissue? If it is, that's a cytotoxic. If it's changing the function, that's a non-cytotoxic type 2. Is it an immune complex made up of IgG or IgM that's circulating? If it is, that's a type 3. And if it's driven or mediated by T cells, that's your type 4. Okay, so that's what we want to do when we're answering these questions about hypersensitivities and autoimmune disease. All right, but I know you got this.